And the first speaker that we will see in this regard, we're delighted that he has accepted to come, is Mark Daly, who's presumably currently in Finland, although Mark has a number of different roles. He's the director of FIM, the Finnish Institute for Molecular Medicine at the University of Helsinki in Finland. So he's probably to some percentage becoming Finnish, but he's also to a large extent still Northeast American, I would say, in his mind. He may say different things to us in a minute. He's a professor of genetics at Harvard Medical School. He's chief of the Analytic and Translation Genetic Unit at Massachusetts General Hospital, and he's a member of the Broad Institute of MIT and Harvard. And for sure, one of the reasons why he got so interested in Finland is that there's a very interesting and relevant population in Finland for those who are not human geneticists. So the Finnish are very special within Europe, fairly small population with that is genetically quite distinct from the rest of Europe. And at the same time, the Finnish have been very diligent in terms of generating a good record of their genetics. And Finland is one of the countries that has actually done very well, I should say, in terms of making genetic data of their population available for research and generating advances for this. And I'm sure that was one of the reasons that inspired Mark to become the director of FIM. Mark actually has been in genetics for a long time. He worked as a freshman already while he was at MIT with Eric Lander at the exciting times of the Human Genome Project. He made important contributions there towards understanding haplotype structures, and then later did really very critical contributions in complex disease genetics for quite a variety of different diseases, leveraging the power of genome-wide association studies and going after diseases such as Crohn's disease, inflammatory bowel disease, autism, schizophrenia, and as we'll hear today, also studying the genetics of complex disease and of complex infectious disease, in particular COVID, which for sure is very complex disease. So he's one of the most cited scientists in the field, and that's certainly evidence for strong success among the 100 most cited scientists of all times. He's an elected member of the National Academy of Medicine, and he won the Kurt Stern Award for his contributions, which recognize major achievements in human genetics. And so, Mark, it's wonderful to have you here and get your perspective on the power of data sharing, and I look forward to your talk.
Yeah, and thanks for the way too generous introduction. I hope I can live up to even half of the advanced press in that paper. It's a great pleasure to be here. I mean, once the invitation came and told what the meeting was and what the topic was, there was no question as to I would be more than happy to come and discuss my thoughts on any of these things. And hopefully, since this is somewhat different than my usual talk, it will be appropriate to the topic and spur some discussion. But it's great to be here, and I'm going to dig into the topic of the importance of open science and data sharing, particularly in the context of common disease and biobank research, and I sort of blend the two together in the talk. My thinking, on this dates back, obviously to my long years in Boston and long before I came to Helsinki, and so I'll start there with the work that we have done in Boston. And me being a little older, I think I acknowledge that I learned a lot from the junior people that I was lucky enough to hire and bring along when I first had the ability to hire faculty about 10, 12 years ago, Ben Neal and Daniel MacArthur in particular. And, we all started thinking together about how the progress in our field as it was emerging through these years, 2005, 2010, was dependent on data sharing and collaboration in a way that we had never imagined it before. And so, pieces like this one by on open science and many others were influential. And what we liked about this particular work was that it made the point that no matter what they call it a school of thought, but I think of it more as motivation, that there are many different motivations, and no matter what your motivation is, it's difficult to see a model where open science and data sharing are not advantageous. And if you think of it in terms of equity and democracy, certainly, but simply in terms of, as you know, is sort of most appealing to some of us with the scientist and engineer hats on, a very pragmatic view, it was beginning to become unquestioned that in a practical sense, we were making much more scientific progress together and on problems that we had made no progress on for many years, decades in some cases, by the sharing and joining of forces with respect to collaboration, data sharing, infrastructure sharing, and scientists and to be funded to do research, because there are many people in the world hoping that we can find solutions to medical problems that are plaguing our world, throughout. In mind, we, were living through a period of time in which there were many paradigm shifts in human genetics, that up until the year 2000 and even 2005, we knew very little, almost nothing, about the actual concrete genetic components of common diseases that were taking the biggest toll on the health economics and the health of our populations. And what we were seeing and living through was a fact that collaborative science, first in understanding the nature of human genetic variation and why diseases were heritable, and then the technical infrastructure were making remarkable strides in giving us opportunities to understand disease for the first time in many cases. But really, I would argue even a more fundamental and important paradigm shift was that human genetics as a field moved from a model of competition to a model of collaboration. And that, of course, required data sharing, and it of course required people to have trust in each other in a way that was not necessarily endemic to the field of human genetics before this time. And so a great example of this has been the progress in understanding the genetics of schizophrenia, and this activity began, probably about 15 years ago, but continues apace to this day. And the latest studies led by Stefan Rübke, who's here in the audience, as I believe, have utilized a hybrid model of data sharing and analysis, and that involves both many studies from countries around the world with the permission and ability and will to share raw data, have done so into a centralized compute server, and Stefan and his team have been most successful at leading the analyses of these data sets to sort of provide the backbone of the activity. But there are many other studies that are unable to do that, and of course then follow the same analytical protocols and share the summary statistics. And as we well recognize, no matter how much we would like to just say, yes, all data should be shared and it should be put on the internet freely and so forth, it's simply not possible if you are, depending on the consent that the individuals were collected on, depending on the country and the interpretation of the GDPR, of course. And in many cases, we also had participation from industry and clinical trial samples that the samples themselves couldn't be shared, but those companies were willing to contribute the data into this joint activity. So this has, turned what was, a a backwater of research in the genetics of psychiatry into one of the most flourishing and exciting areas of biomedical research in recent years. And genome-wide association studies in general have had the success that they've had at identifying genetic variants associated to all nature of complex diseases, largely because of this collaborative model in which researchers throughout the world are willing to pool their resources and perform joint analyses together to advance discovery for everyone. And so reflecting on that, in the time frame about 5 years ago, we made a number of commitments, to sort of try to set standards for ourselves first to follow, to hold ourselves to them. But hopefully, ultimately, if we could manage to follow these ourselves, others to follow as well. And so I think the elements, involve both, posting, any manuscripts on a preprint server so that the results are freely available to everyone as quickly as possible. The one that we've sort of driven the hardest is with respect to the sharing of results and summary statistics publicly at or before the point of submission of any manuscript. And this has, really helped to set a standard and give us leverage in working with other groups to try to convince them to do the same thing by putting the own example out first. Contribution of raw data to restricted access repositories. I think everyone agrees to whatever extent it's possible, it's a good thing. And neither 0.2 nor 0.3 are seen as having any, if they are done responsibly, are seen as putting study subjects under any risk or violating any consents. And then, of course, sharing software, experimental protocols, and generally trying to advocate as much as possible for making data sharing easy are things that, we felt we should be, outspoken in advocating for. So we put some of these concepts into, policies that we put out so that, we would make a public statement that we're doing this. And it was, but, at the time, I felt it was mostly just to, by putting these things out publicly, we would force ourselves to make sure that we did them because somebody else would hold us to it because we said something about it. And I think it's certainly worth noting that, during the last, 5 to 7 years, especially, one of the biggest changes in our field has been the adoption of preprints in a way that it, was ordinary to mathematicians and other scientists, but was, quite foreign to biology, as you can see from this graphic here, but has exploded and really made the biggest difference, I think, in communication of scientific results in a much closer to real time than the standard publication processes allow. So I'll give a few examples of studies and how we managed to, push in the directions of these types of principles, and how the studies themselves, have only been able to be successful because that we've been able to push in the direction of these principles. First study is the large-scale exome sequencing study of schizophrenia. We call it SCHEMA for obvious reasons. This is work led by TJ Singh, who's been a postdoc with our groups in Boston for about 5 years now. And TJ came to work with us specifically on this project. And the model of the SCHEMA project is a centralized data generation model. So much easier in terms of data sharing, because the vast majority of the diverse schizophrenia case control studies were sequenced at one center, at the Broad Institute in this case. And essentially a condition of having your sample sequence was that you would participate in the joint analysis activity to drive schizophrenia discovery. Here's a few pictorial guides, and the study benefits, not only from diverse set of schizophrenia case and control in red and blue, but also in black, shared control samples that are accessible through dbGaP and other restricted access repositories that have general research use and could be employed as additional population controls to increase the power of the study. And that's, I think, a very important, reason why those types of samples that are broadly consented and deposited in restricted access archives are so important, because they can be utilized over and over again to power discovery in many, many unexpected ways. So TJ spent an enormous amount of effort in pulling these data together, and obviously the hardest work was in all the QC, as it is in most of these studies, all the QC and processing and so forth. But he was rewarded by finding, as had been seen in autism and intellectual disabilities, in schizophrenia, a substantial excess of ultra rare loss of function and severely damaging missense variants in schizophrenia cases compared to controls. And those aggregated sufficiently in a number of genes that the first 10 genes were definitively identified by this genetic study. And in the expansion of it that's ongoing now, it appears that number will soon go up to 30 and then 50. And this is, good hard evidence in contrast with generally what we would find from the GWAS, because here we have specific mutations and specific genes with a specific, rather established mechanism of action. And that gives us, a lot more of a concrete biological handle into the biology of schizophrenia than most of the non-coding associations from the GWAS give us. And so while those are more plentiful, they're kind of a little bit somewhat more imbalanced in terms of their contribution to our understanding of the biology. And here's a series of the genes and their potential mechanisms and implicated by the sequencing study thus far. Importantly, I want to come back to the point that, in fact, we, even with exome sequencing study getting down to individual ultra rare variants, we have stuck to the principle that these data must be made fully and freely available to the extent possible. And in this case, the summary statistics and also the summary counts of alleles per gene, even the ultra rare ones, as well as on a variant by variant basis. And this, has, these tools and the making of this available has permitted many, many researchers, both on the sort of functional biology and neuroscience side, as well as other geneticists studying schizophrenia and many other psychiatric and developmental disorders, to take advantage of the data resource. And both these studies and the PGC studies led by Stefan and others have consistently, for the past decade plus, made full summary statistics freely downloadable and browsable on the web. And gratifyingly, a few years ago, after a lot of effort on a lot of our parts, the NIH finally concurred that such data sharing of summary statistics comes with negligibly small, if any, risk to participants, and that any imagined risk, which, as they acknowledge, has never been actually a single time realized, the tremendous benefits to biomedical research and ultimately to the patients are generated from this type of data sharing. And so we are happy that we are on the plus side with the NIH regarding this type of data sharing now, because it does have tremendous value, and I think it underpins our thinking with respect to all common and complex disease studies. So example number 2, now, of course, I'm in Finland since near the beginning of 2018, and a driving reason why I was interested to take on the directorship of the Institute for Molecular Medicine here in Helsinki was that Arno Pelletier and I had designed for many years the concept of the FinGen project, and had only recently in 2017 managed to fund and launch such an activity. So what is FinGen? So FinGen is based around, of course, a program that ultimately has as its goal, generating data and insights that can ultimately be converted into something that might alleviate disease for Finnish citizens and maybe citizens of the world, hopefully as well. And the challenge with building a resource in FinGen, in addition to the fact that these things are expensive and require, some innovative funding, is that, new medicines and medical technologies, for us to actually get to the end point and have some impact, requires industry. Our academic research, however good it is not delivering solutions to the world for, in terms of medicines or medical technologies. However, of course, the progress in industry relies on basic scientific discovery. And what we've had to do is explain to a lot of people that the idea of partnership between academia and industry is not exceptional, but it's required. That if we want to ultimately have impact for human health, we need basic scientific discovery and industrial innovation to work more closely together to advance society's goals. This creates new challenges for data sharing, because of course in Finland we have almost an idealized configuration where the population and the government are very positively inclined towards academic medical research and participating in academic medical research. They have a high trust in this research. And there's nothing more fundamentally important to being able to continue this type of research than to maintain that public trust, and to ultimately get all the way around this circle to implementation of new health. We need to figure out models for performing that research in partnership with industry, but in a way that does not abrogate that public trust. In Finland, and one of the reasons why Finland is seen as a great place to do this type of research, and really what underpins our ability to do a project like the FinGen, is a law that was enacted in 2013, the Finnish Biobank Act. When I heard about this, I couldn't believe there was a place in the world that would actually have a law about biobanking. Coming from the US, it seemed like, boy, you're lucky if you can get a project approved at your own hospital, let alone have an entire country pass legislation about how to do these types of things. But it's a remarkable law that allows individuals to consent to a national biobank framework in which they're giving a broad consent for all approved medical research, without specificity at the time that they consent to exactly what type of research is going to be done. Permitted existing legacy samples, and there are many valuable epidemiologic samples that collections were started 20, 30 or more years ago, could be transferred into this biobank framework. And there's the possibility of recontacting for any specific reason that's, again, approved by an IRB. So these possibilities create a wonderful framework to begin to jump off. But what, in addition to that, is great is that, of course, with the Nordic medical system and the confidence and the trust of the population and the government, there's a possibility to use birth to death health information collected by the government for research purposes. And what that configuration sets up in total is that we can recruit individuals into the FinGen project and into this national biobank framework, and instantaneously have a lifelong phenotype from a variety of national registers that are by law kept and filled by all practitioners, whether it's a public or a private healthcare provider. And so these the foundation that we build on is the National Healthcare Registry System, which actually exists in a number of different registers in different government agencies, which creates some great opportunities for challenging logistics of permissions and so forth. But we have hospitalization diagnoses, cause of death, cancer registries, outpatient and specialty clinic diagnoses from more recent years, and all prescription drug purchases for the last 25 years, as well as for a much longer time in the past, reimbursement codes. And these are all indexed essentially by, largely by consistent ICD systems in all of these different registers. And so they can be combined into not only individual diagnostic categories, but also gives the opportunity to move our genetic studies beyond single time points to longer dynamic views of health and disease. And so the FinGen project was launched in 2017, a genuine public-private partnership. Essentially, we have 13 pharmaceutical companies that pay what amounts to sort of annual subscription fee to have access to the data, but they don't own any of it or anything of that nature. And it's partially funded by the Finnish government through their innovation arm, Business Finland. And it's a partnership involving all hospital biobanks throughout the entire country. And as you might imagine, given especially the fact that this is based around a national biobanking law with very strict requirements, and the fact that it is utilizing the individual sensitive health information of Finnish citizens and ultimately aims to study 10% of the entire population, security and consent are the first priority that need to be taken care of in as rigorous a way as possible. And that's because, as I said, we have largely a middle-aged and older population, and their entire health history, or quite much of their health history, depending on how old they are, is contained in the events that we capture and can utilize for analytic purposes in the project. And so with this level of sensitive data, we had to construct, obviously, an extremely secure environment with which to perform research activities utilizing that data. So this is never going to be, obviously, a data set that can be shared broadly in any sense of that word, as it's not likely that many of the sort of health system data sets that have intimate data from health registries and records can be broadly shared. So what we've constructed, and we're fortunate to get approved, was a sort of a, before the term became, more popular, a trusted research environment that we call a sandbox that's implemented on the Google Cloud. And trust me, this took a lot of convincing to get, a Google Cloud, instantiation approved for use, as all the natural questions, sensible or not so sensible are asked of it. But allowing us to have a virtual environment that can access and run computing jobs on the sensitive individual genotype and phenotype data, but not allowing that data to be copied out. And I think those of you with a sort of computational hat on who are thinking about these issues, this is a fairly now straightforward configuration. That sandbox, as I said, contains obviously all of the individual level data. It's pseudonymized so that no individual identifiers are contained there. But of course, according to GDPR, this cannot be considered anonymized data whatsoever because it is re-identifiable. And happy to have join any long debates about the questions of re-identifiability versus identifiability of data and what we should be doing in each of those cases. But the infrastructure essentially allows individual partners, be they academic or industry, and whether they're located in Finland or in the US or wherever, to have their own private workspace that allows them to connect to the FinGen data and run jobs on their own. And we add to that, obviously, all of the standard tools that make it, straightforward to run your own jobs, as well as a number of custom utilities that we've built to make running, standard GWAS jobs straightforward, reviewing and editing, genotypes from the genotyping arrays and so forth. The nature of the data access, it comes in sort of two pieces. One is, it's not necessary that everyone has this individual level or red library access to the project, because only a handful of people actually are interested to run their own jobs on the data and really dig into the questions that require access to the individual level data. And so the majority of users actually have a higher level of access called the green library access. And this allows you access to all of the in progress summary statistics and analyses that are run, and is a much easier access for most people to work with in terms of what they need to get out of the resource. And ultimately, the things in the green library are things that become public after a period of time, or can become public if anyone's interested to have them. So the project, as I said, was launched in 2017. And really, I think the thing we're most pleased with is that, despite the fact it's an industry-funded project, we've, the Finnish participants have benefited tremendously, and we've had no resistance to making the data freezes periodically publicly available. And so any of you can log in right now from the auditorium and go to this FinGen website, browse the genetic results for thousands of pre-calculated diagnoses, thanks to Mitya and Juha in particular, including novel genetic discoveries for anything, in this case, inflammatory bowel disease, and using enhanced versions of FeeWeb tools, look into fine mapping, co-localization with quantitative traits from UK Biobank, or expression QTLs from the eQTL catalog, and then browse the entire span of phenotype associations for any genetic factor you might be interested in. And we're also particularly happy this year that the data became was fully loaded into the Open Targets toolkit, and is now a major part of when you browse the Open Targets toolkit, you get all of these FinGen results as well. But probably the most impactful part of sort of being as open as possible with this type of resource, especially with respect to the sharing of the summary statistics, was that it allowed us to sort of set an example that helped as leverage, and helped as in initiating a large number of other multi-biobank, multinational efforts. Now, of course, the UK Biobank is uniquely leads in this area by having a remarkable consent model that enables the distribution of the data broadly around the world. That may not be a model that anyone else will actually ever be able to necessarily replicate, but we can hope. But certainly for collaborative activities, I think we have now entered an era where biobanks are becoming more common and being built up in many, many different locations in the world. And this idea that at some level, we need to work together and collaborate to build larger multinational efforts is starting to take root. And so we can see the results of this already starting to come forward in a number of different studies. Masa in the group in Boston has led some nice example work, which is available on medRxiv now, demonstrating that when you begin to perform joint analyses of biobanks, in this case, UK Biobank, Biobank Japan, and FinGen, you can start to see much more compelling evidence of human genetic involvement in a number of phenotypes. And what he's showing here is a number of genes implicated with 4 or more distinct coding variants across these 3 biobanks. And so diversity of biobanks is now yielding much larger sets of variants in each gene and allowing us to make much more durable inference from human genetics based on that. With those early experiences, 2 years ago, we launched a global biobank meta-analysis initiative, and this has had great participation from now about 20 or so biobanks around the world. This was featured in a number of talks last week at the ASHG meeting, and I'd encourage people to take a look at that if they're interested. And the goals of this activity, and this is purely in a model of meta-analysis where each group is performing its own biobank analysis and then sharing the summary statistics and computing them using a common and agreed upon set of protocols and methods, and then sharing the summary statistics to a common server for joint analysis and discovery. And the rather obvious reasons of improving power and resolution, but especially increasing the ancestral diversity and what we can learn from that are really at the heart of this activity. And it's been extremely gratifying over the last 2 years to see just how many biobanks are willing to collaborate at this level, which is not overly strenuous, not giving a lot away from your own research, and certainly not compromising any data security, but a very productive activity nonetheless. And then finally, I think many of you are aware of the effort that Andrea and I launched now 1.5 years ago, which got, again, an even more remarkable response from research groups around the world to perform a single, collaborative effort to approach the host genetics of COVID-19 susceptibility and severity. And I won't go through, too many of these slides in great detail, but at this point in time, and sticking especially to the core mission of this activity, which was as soon as an analysis is performed, that's a joint collaborative analysis across biobanks, it's simply posted to the web, because anyone anywhere might be able to make use of it. That would be fantastic. And at this point now, 125,000 COVID-19 positive cases have been studied. The samples break down quite evenly among established biobank studies and newly initiated studies in clinical sites and hospitals, specifically for the study of SARS-CoV-2 infection and severity. And there's been, now a good set of genetic discoveries, more on the severity side than the susceptibility side, which, given the remarkable, the emerging, remarkable observations of how much, asymptomatic COVID versus life-threatening COVID there is in the population, is probably not surprising that that's where most of the weight has ended up. There's very interesting dissections of certain loci in which there's both a susceptibility and a severity locus independently sitting within 100 kb of each other, and the strongest region of association on chromosome 3. And so, using Bayesian methods that my colleague Matti Pirinen down the hall has developed, despite the noise in all of the studies and the very heterogeneous ascertainment of patients from biobanks and hospitals and direct-to-consumer studies, we could fairly convincingly distinguish each association as being a locus attributable to susceptibility or a locus attributable to severity as deriving its effect. And in the interest of time, I won't walk through all of the results. Some of these are published in the first consortium paper that emerged in this past summer. Some that are the most interesting to me anyway are a locus at FOXP4 that really exemplifies a discovery that required diverse populations beyond Europe to really make the genetic association convincing, but also leads to a very interpretable finding in which the variant clearly increases COVID severity, clearly has been previously associated to lung cancer and interstitial lung disease severity as well, and increases the expression of FOXP4 in the lung. Similar nice tidy story with respect to a tyrosine kinase on chromosome 19 that's been enormously well studied as a source of potential therapeutic for autoimmune disease, whereas in the opposite direction, the hypomorphic allele is seen as promoting COVID risk. So might make you cautious about inhibiting TYK2 in the presence of a pandemic. And then a few tidbits from the latest study, which is available on the web but not yet published, a surfactant protein with a previous role in lung disease. The strongest risk factor for idiopathic pulmonary fibrosis is actually strongly protective from severe COVID-19, which is quite interesting and unexpected. And what was actually much more expected, genetic variation at ACE2 is clearly associated with susceptibility or protection from infection with respect to SARS-CoV-2. So the goal of these activities, coming back to where we started many, many years ago, is to ultimately do things that have some impact or help for our citizens. Hopefully, I think I've convinced that robust discoveries in human genetics really require collaborative studies, which require, in turn, data sharing in as rapid and open a way as possible, though we have to be flexible in these different types of studies with different models. And these discoveries are really now beginning to take off and being taken up in academia and industry. So thanks very much. And thanks for all of my colleagues in the FinGen project and many of the other studies that were mentioned as well.